Hello everyone and welcome to episode 15 of CCLG's Research Talks for Parents and Public. I'm Ellie Ellicott and my job is sharing research with our supporters. So starting with some housekeeping, we will be holding a Q&A session at the end of the talk, so please do submit your questions whenever you think of one using the Q&A button that should be at the bottom of your screen. If you'd like to use the chat function, make sure you've set the to drop down to everyone or people may not see your message. The webinar is being recorded and will be available publicly on our YouTube channel, so I'll send out the link next week when it's ready to watch. This month's talk is called Our Journey into Childhood Leukemia Research and is being led by Doctors Cristina, Pina and Sabrina Tosi. Cristina and Sabrina work at Brunel University of London and lead a Little Princess Trust funded research project investigating targeted treatments for babies with acute myeloid leukemia. So, without further ado, I'm going to hand over to our speakers for tonight.
Hi, let me share the presentation so that then I can introduce ourselves. And 1 second. Okay. Okay, is it visible?
Perfect.
Okay, so, both Cristina and I are very happy to present our research to the public, and we thought we would start with a little introduction on ourselves. I graduated in biological sciences at the University of Milan, Italy, and then I specialized in human cytogenetics, which is the specialty, the study of chromosomes. I was quite lucky to start my work on leukemia quite soon after graduating in a hospital, in a pediatric clinic specialized in hematological disorders. And this is when I started my journey and my interest in the study of childhood leukemia. After that, I spent some time in a kinder clinic in Germany, a year for a research experience. And then I came back to Italy, and after a while, I decided that I wanted to further my investigations again with an experience abroad, and I then spent 12 years at the University of Oxford, where I did my DPhil, and I did a lot of good things, some of which I will talk about during this presentation. What I am particularly proud of is to have identified a very rare form of leukemia that affects very young children, and this is going to be the topic of our presentation. Cristina?
Hi, so my name is Cristina Pina. I'm originally from Portugal, where I trained as a clinician, and I was a clinician for a few years until I decided that my passion was doing research, and I dedicated my professional life to that. So I came to the UK in 2003 to the same institute in the University of Oxford where Sabrina was, and I think we maybe coincided for a very small period. I did my DPhil there, and then stayed a little while for a postdoc, did another couple of postdocs at UCL in Cambridge. In Cambridge, I started my independent group and then came to my first substantive academic position at Brunel 5 years ago. So I've always been interested in blood formation, maintenance and differentiation of blood stem cells into all the different types of blood cells, and very particularly in how that those decisions that cells make are regulated at a molecular level. And inevitably, that led me to looking in detail into processes of leukemia initiation. And in the last 7 or 8 years, my lab has been interested amongst studying those molecular mechanisms in building models of blood formation as it happens in the embryo, and I'll show you a little bit of that. And in that process, I became quite aware that that gave us a window of opportunity into understanding the initial events of leukemic transformation in leukemias that affect very young children. And being both at Brunel, Sabrina was the ideal partner to further that research. And after a few years of collaborating informally, we've joined forces, and we are very grateful to CCLG and the Little Princess Trust for enabling us to do that. So maybe we'll just tell you a little bit about the work we do, how we came to it, and then, of course, we are delighted to answer your questions and just talk a little bit more informally about our research and our work. So Sabrina is controlling the presentation. So this is sort of back to my interest in understanding how the different blood cell types are made, how they are made from stem cells and progenitors, and what controls those decisions. And very broadly, there are more than 12 or 14 types of cells that are made from blood stem cells. These are the ones that are most abundant. Red blood cells, of course, that are responsible for oxygen transport to the different tissues. Platelets involved in clotting. Lymphocytes, which are our immune cells and are the non-specific defense cells, that of which the most abundant are neutrophils, but also include monocytes and other cells that are generically known as granulocytes. And amongst the cells that form the white blood cells, which include, amongst others, the neutrophils and the lymphocytes, reside the main types of leukemias. So there are leukemias, a group of leukemias with some common characteristics that affect lymphocytes, and those, in the case of leukemias in children, are typically lymphoblastic leukemias. And then the cells that affect the leukemias that affect the other white blood cells that are not lymphocytes and can also affect precursors of red blood cells and of platelets, are known as myeloid leukemias. And fortunately, leukemias are rare. They are rare in children. And currently, they, particularly in the case of the most frequent leukemias that once diagnoses in children, which are acute lymphoblastic leukemias, they have very good prognosis in the large majority of cases. Acute lymphoblastic leukemias, particularly acute B-cell lymphoblastic leukemias, are more common in children and adolescents, and that is the opposite pattern to acute myeloid leukemias that are a lot more abundant in, a lot more frequent in older people. However, in the first year of life, they are rare at 15 to 20 cases per million, but they are almost equally frequent. And we know quite a lot about acute lymphoblastic leukemias in children, and that is in large part because they are more frequent, and in large part the reason why it is there are more treatments and there's more knowledge about them and there is better prognosis. We know relatively less about acute myeloid leukemia in children. So in the adult, the precursors of myeloid cells and in fact of other cell types reside in the bone marrow, so in a specialized tissue in the cavities of some of our bones. That is where they reside, that's where the stem cells reside, and where blood is produced throughout life, and where leukemias are initiated and diagnosed. However, that is not where blood cells reside during embryonic development, during the development of the embryo. In fact, the way in which blood is made in the embryo is very complicated, and it uses multiple locations throughout the first trimester of human embryonic and then fetal development. And in those different locations, there are different types of cells that are made. The very last ones that are made are actually the stem cells that are going to sustain the production of blood after birth and throughout adult life. And what is important is that some of the precursor cells that produce blood in the embryo and in the fetus, and which are distinct from stem cells, actually are not present in the bone marrow and in the blood system after birth. So they are relatively short-lived, they are transient, they are very characteristic of the embryonic development, and they do not persist after birth. And the reason I am calling your attention to them is because at least in some forms of leukemia, including we think the one we study in more detail, that is important and they play an important role. And so when a few years ago, we started making 3D models of blood formation, trying to capture all these different phases of blood production as they happen during embryonic development. And we do this in these multi-well plates with small numbers of pluripotent stem cells, either mouse embryonic stem cells or human pluripotent stem cells. And we use minimal growth factor cues and to guide the assembly of these 3D structures that for the most part self-organize with a little help from the culture conditions. And we are able to recapitulate quite a number of the processes that I was showing you in the previous slide. And that illustration on the right is a view of one of our mouse 3D models. We call them gastruloids, or blood-forming gastruloids, and where you can basically see something that we also see in during the formation of the blood cells in the embryo, which is the blood cells emerging from a specialized endothelium, which is the lining of the blood vessels, in little clusters that bud off from the endothelial cells. And so the endothelial cells are shown in green, and the clusters is of the progenitors, the more immature cells, and the mature blood cells are what we see in yellow and in red. And the blue is just the nucleus of the cells, so that we know in the under the microscope where the individual cells are. And so we were able to, using this, we're able to capture a lot of these transient cell types that are also detected during embryonic development and don't persist in the adult. And the reason why at least some of these are important in understanding leukemia is because it has been known for quite some time now, in a large part through the work of Mel Greaves and collaborators, that there are a number of leukemias of children that start in utero, so during the development of the embryo. And this was shown very elegantly many years ago by the detection of the mutations, that the rearrangement of the chromosomes that Sabrina is going to talk about in these the blood from these heel prick tests that the babies do to detect a number of genetic disorders shortly after birth. And so it was well established that some forms of lymphoblastic leukemia common in children originate during fetal development. And it has actually been shown more rarely, but it has been shown for a couple of acute myeloid leukemias that is also the case. And one of those myeloid leukemias is the one that we're interested in studying, which is the one that is very unique to children under the age of 2, and that is driven by a chromosomal, an exchange of chromosomal material that Sabrina is going to tell you about.
Okay, I think I lost a connection for a few seconds. I hope this didn't impact on the presentation.
No, that's fine.
Okay, so yes, here I'm talking about chromosomes, because this is what I specialized. I have been trained as a cytogeneticist, so I'm a scientist, a chromosome biologist, essentially. So you all know that the genetic information reside in the DNA that is contained in the nucleus of each cells of the human body. And the DNA is condensed in corpuscles that are known as chromosomes. And these are visible under the microscope at a certain point of the cell cycle. So when the cells are in division, the DNA condensed to form this little corpuscle that are the chromosomes. And as you can see on the top right part of the figure, this is called a chromosome spread. So when we prepare the cell samples in a particular way, then we are able to visualize chromosomes under the microscope. And then what cytogeneticists do, they pair the chromosomes and they identify the different chromosomes according to their binding pattern. So we have a pair for each chromosome because we inherit one from the father, one from the mother. And as you can see in the bottom right picture, this is called a karyotype. So when we align the chromosomes, we pair them and we align them in order of size and binding pattern. So this analysis, the analysis of a karyotype, helps us understand if there is any genetic error that is, at a scale big enough to be identified at the chromosomal level. Okay, so what I want to say now is that actually, leukemia is cancer of the blood, and it involves abnormalities of the white blood cells, and it develops when the blood cells undergoes transformation into malignant cells, which is capable of uncontrolled growth. So we say that leukemia is a clonal disease because when there is a genetic abnormalities happening in one of these precursor cells, then the daughter cells will inherit the same chromosomal abnormalities. So there will be a generation of identical cells that carry the same genomic asset. And these genomic abnormalities are often visible as chromosomal rearrangements. So there are a number of chromosomal rearrangements that we are able to identify, and this can involve exchange of chromosome material between different chromosomes, or loss or gain of chromosome material. And here is a picture of a 24-color karyotype. Well, not karyotype. We mastered a technique that allows us to color code the chromosomes. So it is already useful to have the banding pattern as we see on the right-hand side of this slide. But with the technique, we say fluorescence in situ hybridization, we can use specific fluorochromes to identify specific chromosomes. So this makes it easier to see whether there are exchanges of chromosome material between one chromosome and another. And this is an example of a 24-color karyotype that is generated from the cells, the leukemic cells of a patient with acute myeloid leukemia. As you can see, chromosomes are quite messed up in cancer, and especially if there are a lot of chromosomal rearrangements, this means that the cancer is a quite advanced stage. And this is what happens in solid cancer, colon cancer and ovarian cancer. We have a lot of abnormalities. In adult leukemia, we, depending on the stage of the leukemia, we can have a lot of chromosomes involved in abnormalities, as you can see in this picture. In childhood leukemia, normally we see very straightforward, simple abnormalities, unless at relapse we can see additional abnormalities, sign that the clone is evolving. Okay, so and this takes to the talk to this particular chromosomal translocation that we mentioned at the very beginning of this presentation, which I'm very proud of, because when I was at Oxford studying for my DPhil, my project focused on abnormalities of chromosome 7 in acute myeloid leukemia. well, not just acute myeloid leukemia, but leukemia in general, hematological malignancies in general. So these were the days when this technique of fluorescence in situ hybridization came into the labs. And I was one of the first to use it to study leukemia in those days. These we're talking about the 90s, so this tells you how old I am. So during the course of these studies, I happened to see an abnormalities that you can see here between chromosome 7 and chromosome 12. So in this picture at the bottom, we find chromosome 7 in green and chromosome 12 in pink. And these techniques allowed us to identify very, very small regions of the chromosome translocated on a different chromosome. In fact, here you can see indicated by the arrow chromosome 7 with a tiny little bit of pink that is derived from chromosome 12. And the translocation of chromosome 7 on chromosome 12 is so small that actually you can't see with this technique that is called chromosome painting. But then we refined using other probes that are smaller and more specific for the chromosomal region. We managed to identify exactly the regions involved on both chromosomes in this translocation. Interestingly, during these very early studies, I found that, first of all, I found one patient with this translocation. Then I happened to find a second patient. And then we say, when you find a rearrangement in at least 2 patients, this is no longer a random abnormality. And this is very important from the clinical point of view, because then you start looking at similarities between among the patients. And we found that these 2 patients were both babies, so very, very young children. They were both under 2 years of age. The leukemias were both myeloid leukemia, were both acute myeloid leukemia. So we continue investigating in this area, trying to collect more samples from our collaborators. And I want to emphasize in this talk that actually collaborations in research are really, really important. And thanks to the connection with different hospitals, we were able to collect what is considered the largest series of patients with the 712 translocation. And I'm talking about like 7-10 patients. So in the whole world, the number of cases is very limited. So these samples are really very precious. So we associated this particular chromosomal abnormalities with this specific series of young babies with acute myeloid leukemia. And when we went to look at the clinical outcome of these babies, unfortunately, the prognosis seemed really poor, with very poor survival time. And this prompted us to keep studying these patients, because the ultimate aim of cancer research is to find therapies that can actually be targeted to the specific type of cancer. And by studying the chromosomal abnormalities, by studying the genetic asset of the different type of leukemia, we can find out what are the biological mechanisms that are different in different types of leukemia. So when we find the biological mechanisms of the different type of leukemia, then we can think of targeted therapy that can actually resolve the disease. But going back to talk about the 712, what we found is that because of the transfer of material of chromosome 7 onto the chromosome 12, this mispositioning of genomic material results in a gene activation of a particular gene that is called MNX1. What is interesting about this gene activation is that MNX1 is a developmental gene. So it is one of those genes that are called homeobox genes. They are active during the embryonic development. They have their job to define orientation of the individual, development of different organs, but then after embryonic development, they are silenced. But what we found in this leukemia, because of this translocation, MNX1 gets activated again. And we find that all babies with this translocation in their leukemic cells have overexpression of this gene. Now, one thing I want to also emphasize is that when we talk about chromosomal abnormalities in leukemia, these abnormalities are only seen in the bone marrow or in the peripheral blood when we have a high number of malignant cells. But the genetic rearrangement does not affect other cells in the body, affects only the cancer cells. And in this case, the cancer cells are the myeloid precursor of the blood cells. So the way we took the project forward after the identification of these abnormalities, and because, as I told you, these patients are so rare, and being infant, you can't really get a lot of bone marrow to be able to study the samples for a long period. And unfortunately, because of the poor clinical outcome, it was not possible to have second samples from the same patients, for instance. So what we decided to do, we used gene editing technology to reproduce the translocation in vitro. We managed to do that. We now have a cellular model of this translocation that is a cell line. And the cell lines are very important in research because they can grow for a long time in the lab, and we can freeze them, we can use them again, we can put chemicals to see how the cells react. So we managed to establish a model for infant leukemia that has this particular chromosomal translocation. And the aim of our research is to actually use this cellular model as well as other models that Cristina will continue to describe to find a cure for this deadly form of cancer.
Okay, so this is where we joined forces at the end of the or towards the end of the engineering of and the analysis of that engineered cell line, and we were fortunate to get funding from CCLG through the Little Princess Trust to actually be able to carry on research using that model for drug discovery. So what we do is we basically combine computational methods that allow us to triage from thousands of drugs to identify the ones that are more likely to be active in against cells with this chromosomal abnormality. And then we use the cell line to test their effect. And we use a variety of different assays, of which I am showing one that is sort of visually more easy to describe. And that is based on the principle that even in cell lines, the cells that maintain the leukemias growing are actually, in most of the leukemias, a small number of cells that have stem cell-like behavior, even if they are not necessarily stem cells themselves, but are essentially the ones that are able to maintain the leukemia long-term. And these are very important because these are ultimately the ones that need to be targeted by any effective drugs for the leukemias to be eliminated. And in order to screen the drugs that we had already shortlisted through computational methods, we basically made use of this engineered cell line and put the cells of the engineered cell line to grow in a semi-solid medium, like a thick gel, where the cells grow separated from each other. And the ones that are able to grow a leukemia form these little colonies of cells in this thick gelatin. And this is what you see in the panel on the left, where the cells were allowed to grow in these cancerous colonies, and they were treated just with the carrier that we use to dissolve the drugs. And so these cells form colonies, but in the center and in the right panels, we're showing you two of the drugs that look more promising in our studies, particularly the second one, where it is clear that there are less of those, colonies growing, and so suggesting that these drugs are actually effective in eliminating, either by killing or by preventing the growth of those cancer leukemia initiating cells. And so we confirmed the potential interest of these drugs with other tests in the cell line. And then we thought that it might be interesting to go and test these drugs at mimicking the very early stages of the development of this leukemia, which we know from studies from colleagues in Spain whom I collaborate with in some other projects, that has an origin during embryonic development. And so what we did was, we are still in the process of recreating the translocation in the pluripotent stem cells with which we make our 3D models. But to start the work, what we did was we took advantage of the fact that this chromosomal rearrangement always leads to abnormal high expression of this gene MNX1, and we recreated that abnormal high expression in our 3D models. We did see, we had results suggesting which of those transient cell types that we only find in the embryo is the likely origin of the cell, and we still don't have a definitive answer, but we have very good indications, and we're following that up with other studies. But what we also saw was that if we treated these 3D models that have this abnormal increased expression of MNX1 with the same drugs, we actually saw a reduction of these candidate cells of origin of this leukemia. And these cells are amongst the cells, are a rare population of cells, but are amongst the cells that we are here coloring in green with a reporter on a molecule that marks these very early blood cells in the embryo, but also in our 3D models. And what we can see is that if we treat these 3D models for a very short period, which is the period where we think those critical cells are being formed with our drugs of interest, particularly with this drug 2, we actually see a reduction in those very early blood cells in our 3D models. And if we collect the cells and then treatment like we did in the engineered cell line in these same cancer colony forming assays, we actually see an almost complete elimination of candidate cancer initiating cells when we treat with this drug too. So these are very exciting results that we're still characterizing further to understand the mechanisms. And we are starting to, for now, still in our laboratory models, to combine both these two drugs, but also these drugs with drugs that are commonly used to treat leukemia with variable success to try to suggest a likely therapeutic route for better success in the survival, the prognosis, and the survival of children with this very specific form of leukemia. And of course, it is not really us that do the work in the lab. In fact, I think the people we work with would be very happy for us not to be in the lab at all. And these are actually the lovely and hardworking, very clever people that work with us in the lab. And you have their names there. And I think we keep them happy enough because they are all smiling in the photograph. And the people that have contributed the most to different aspects of the project that we showed you today were Denise, who engineered the cell line when she was doing her PhD, with Sabrina, and at the late stages with me as well. And then Emily, Ilenia, and Iona have made huge contributions to different aspects of the drug screening and the 3D model. And these are all the funders that through the years have funded different aspects of mine and Sabrina's research, with a big thank you to CCLG and the Little Princess Trust. And thank you to you all for listening.
Thank you very much for your talk. It was really, really interesting to hear more. I am trying to get in my mind straight what to ask first. So I think if you wouldn't mind stopping sharing your screen, Sabrina, then we can look at your lovely faces, much bigger. We'll start with a question from Dr. Duff. He's asked whether you use any AI tools in analysing your data or drawing conclusions from your studies.
So we could, and we are actually looking into doing that, because I think if we can do the sort of analysis we did at a much larger scale with sort of machine learning models, we did a sort of a simplified version of that, where we made use of available databases that show us the drug sensitivity of a number of different cell lines representative of leukemia as well as other cancers. And we looked specifically at the combination of the response to drugs and the expression of genes that are very characteristic and that we indeed also defined very characteristic of this form of leukemia. And out of statistical combination of that information, Ilenia, who was in the photograph, came up with a list of the most likely targets, and those are the ones that we've been using in our screens in both of the cell models. But of course, you could, at a much larger scale, you probably could pinpoint it and further it down the sensitivity and the specificity of the results.
Thank you. So this is a question from me because it's something we've talked about before. But how are you wanting to get parents involved in your work?
Do you want to answer that?
Yeah, well, I think communicating to the parents is very important because although both Cristina and I had experience in working in the clinic, and when I was in Italy, in the pediatric clinic, I could actually see these young patients suffering. And I remember also interacting with the parents, and we have a lot to learn from their own experience, because our experience is from the lab, from the science point of view. And we really want to establish a better communication so that we can, first of all, address their curiosity about what's going on in the labs, right? But also we really want to understand what are the expectations from parents, from people that are involved on a daily basis dealing with a horrible disease. What can we do? What are the expectations from the public? What are the questions we need to answer? So initially, I think it's very important to establish a better communication, because I think we tend to, as scientists, we tend to, do our own research, and we really want a better connection with the reality, of the patient. Yeah.
I would just add to that the other thing that would be really great for us would be if not only if when we come to decide what are the next steps of our research, obviously within the capabilities of what we can do, it would be lovely to hear your views of what you feel is important to be the next step of the research, and also get your critical view of actually how well we are communicating it, because a lot of sometimes the weakness of great research projects is that if we can't communicate them in a way that they are understandable beyond the scientific communities, their impact is greatly reduced. So we would be very grateful for your thoughts, your priorities in terms of future research avenues, but also your thoughts and your criticism and your suggestions in terms of how best to communicate what it is that we actually do.
So I can send out your email addresses and a bit of details for all of the attendees. So if anyone fancies signing up and helping out, then you'll have the details. But we have had another question. So how soon are you able to look at the possible side effects of these drugs if and when they are used to treat patients, or if and when when you'll be able to?
Not all of the drugs, but a large proportion of the drugs that we are screening are drugs that are already approved for other uses. Not all necessarily tested extensively in pediatric patients, but they are at least drugs that are already used for other conditions, so they have passed the safety criteria, and so our intention is to try to repurpose them if they indeed prove to be useful for this particular form of leukemia. And that accelerates, although you would still need to go through clinical trials if they prove strong at a preclinical level, but it would shorten the path to clinic. Not all of the drugs that seem interesting to us are approved for other uses, but some of them are, and we would, where possible, prioritize those, because then the path to actual translation becomes shorter.
Thank you. And we have had another question. So would the drugs be used in combination with other modalities, e.g. radiotherapy?
Yeah, children with leukemia are primarily treated with chemotherapy, some very aggressive schemes of chemotherapy, and any new drug that is introduced tends to be initially introduced in combination with those chemotherapy schemes. And some of the experiments we're currently doing in the lab with some promising results indeed are in combination of these drugs with the drugs that are currently used in chemotherapy.
Sounds quite exciting, especially if it's the repurposed ones that are doing the best, because obviously that hopefully cuts down on a bit of the time. I don't think we have any more questions. I don't know if there's anything either of you would like to say before I do my end thing.
No, apart from thanking the people who attended and asked questions, and we very much look forward to hearing your views and suggestions, and do come and visit us.
Yeah, this is what I was going to say. We would be happy to show the environment where we work and all the facilities that we have at Brunel, and we would be happy to have conversations in person if possible. And to conclude, I really would like to thank Ellie and CCLG for giving us the opportunity to present our work in this setting. So thank you very much.